here on this vessel, and this is Kurt Raythorn.
How's it going? Well, I'm actually an engineer, so I maintain all the mechanicals. I work on the plumbing, the electrical, the generators, the main engine. You know, anything that breaks down, I have to know how to fix, or my boss knows how to fix, and we work together on those things. So science was one of my favorite subjects growing up, and I was always kind of mechanically interested and inclined in that sort of thing. So this was kind of something that I felt suited to.
Nice deck. Everything's secure on deck. Hi everyone, this is Nick Terry on the back of the Endeavor on the fantail. We just brought in the wire flyer. It was out in the water for a few hours and we brought it up. It's a whole team effort to bring it in. They reel it, they bring it up with the winch, get it up to a certain height, and I actually got to help this time. I moved the A-frame up to bring the wire flyer and the clunk weight in. You move it with the control panel and the whole A-frame shifts all the way vertical to bring everything on board. And you get to communicate with the bridge, so I was talking with the bridge, letting them know what was going on, so they have an idea of what's going on back on the fantail. And then when everything's set to travel, the A-frame always rests back in the out position, and so I got to move the A-frame back out. So it's a whole process to put the wire flyer in and out of the water, and it was a fun experience getting to help out with the team.
Hi, I'm Trish Garland. I'm an advisor at the East Bay Met. Here we've got the CTD, or Conductivity, Temperature, and Depth sensor system, and we're going to be deploying this. We've deployed it probably about 6 times already on this cruise. And so basically, right now we've got a couple of teachers opening up all these canisters on here, and that's going to get lifted up on this arm, put out in the ocean, and then as it goes down, water runs through it, and we can close it off at any point and collect water samples from different depths. And there's some sensors on there too that are going to pick up temperature, oxygen, fluorescence, and salinity.
Hi, my name is Meredith Ashworth. I teach at Narragansett High School, and we're just preparing to launch the CTD. So safety is a big factor, obviously, on a ship. Yesterday we had a lot of water coming aboard and whatnot, so right now we are just putting the lines on that we're going to use to launch. So we've learned a lot about tying the ropes and whatnot. So there's 2 that will be used to launch, along with the winch operator. And soon you'll see the technician from the boat, Lynn, come over, and she's the one who's in charge of communicating with the winch operator right above us. We'll take care not to stand above directly below the line. And then Joe and I will be releasing it with these 2 tag lines. So this we just leave loose so that we can keep letting out some slack as they need it to release. And then when we do recovery later, it'll be just the opposite. We'll be pulling up the slack. And then right now, they have it pretty secured on there.
Hi, I'm Joe Bodasevich. I teach at Kickman Middle School. Anytime before we move something off the deck, we have to make sure it's still secured. So if a gust of wind comes up and the wave rocks the boat, it doesn't move and injure people. So we use these tie-down straps, these ratchet straps, to keep everything in place until we're ready to lift the package over the side.
Hi, I'm Alyssa Wood. I teach science at Sophia Academy.
And I'm Kurt Rethorn. I'm an engineer on the research vessel Endeavor.
So we're going going to get to do a really cool activity with the CTD. We're going to be taking styrofoam cups, and we're going to be looking at a particular property of these cups. So styrofoam is not very dense. It's actually filled with pockets of little closed systems of air. So the idea is that we're going to attach the cups, we've all got to decorate them, to the CTD and lower it down to the depths of the ocean, about
2,500 meters.
2,500 meters. And we're going to see what happens. So what we think will happen is that all the pressure, all the mass of water on top of the cups will cause the air inside to compress, the molecules get closer together, and shrink our cups down to a very small size. And I'm going to talk about this cup.
Well, this one's for my mom, obviously. And on the top I have our coordinates and the depth. It's 39.5 north, 70.5 west approximately, and 2,500 meters.
So we'll see what this cup looks like on the other end. All right.
She want to get us, Lucy, she want to get a game around this. Do you want to get me to take the ratchet off?
Does it say Mom?
What I really want to try and see is that
All secure.
Today for dinner we have shrimp and sausage, chicken jambalaya, beef tenderloin, rice, potatoes. We get spoiled here. On this boat, we have had some exquisite meals. Chef Amanda has outdone herself. For breakfast, we had standard eggs, bacon, sausage, delicious. For lunch, she surprised us so many times. This afternoon, we had crab cakes. And for dinner, just now, I had some shrimp and chicken jambalaya over rice. And right now, I'm between dinner and dessert, and I cannot wait. The meals here have been fabulous. They have been very, very filling. Not what I expected on a ship. And the chef crew has also been so nice. They also gave us snacks. They left snacks out after dinner, after lunch, after breakfast, all the time. Some of the snacks range from candy bars, just Twix, to muffins that she baked. It was delicious. Overall, I would rate this food on this boat an A+.
Hi, my name is Jessica Grant. I am a 4th grade science teacher at Blackstone Valley Prep. As you saw outside, we just deployed our CTD machine, and it's going to a depth of 1,000 meters, and we're going to collect some samples on the way back up in those bottles that you saw. So if you look at this graph right here, each one of these lines it means different things. So the green one is all about how much chlorophyll is in the ocean. So you can see there was a big spike just before we hit 100 meters. So right there, that until the chlorophyll has been growing all day long, that's why you see that spike in the green. And then the purple is oxygen. The red is temperature, so clearly as we go into a lower depth, the temperature goes down, it gets colder. And then the blue is salinity, so how much salt is in the water. So that's what the graph means. When we get to 1,000 meters, we're going to fire the first bottle to capture some water at 1,000 meters, and then we're gonna have them bring it back to the surface. 2,000 meters, 2000. Doghouse lab, go on down to 2,000 meters, 2000, please. Do I focus so you heard me? Doghouse lab, come up to the surface, please. Cool. Woo!
Hi, I'm Lynn Butler. I'm the ship's technician on this trip. I am going to remove the styrofoam cups from the CTD package. We sent them down to 1,000 meters and they shrunk, and now we're going to remove them and see what they look like.
Hi, we're here back at the CTD Rosette, and Kurt is fishing out his cup for Mom. See it?
Yep.
All right. So
I've got my hand on it.
here's our control. So let's not forget what it looked like before last time we talked to you. And there is the new same cup.
Side by side.
And so just to give an idea of what happened, we learned recently that if you think about the pressure of one atmosphere, that's equal to the pressure given by 10 meters of water if an object was placed under 10 meters of water. So it's as if this cup, well, it actually happened, was placed under 101 atmospheres of pressure. So that's what caused that shrinkage in that cup. So there you have it.
So now we're collecting a water sample. Each bottle is fired off at a different depth, as you saw Jessica show you inside. So in order to collect the sample, we first just loosen the nozzle, and then as soon as we let the air in up here, it should come out. She's gonna clean out the bottle for you a little bit. So, a little failure on fire. There we go.
So we usually do 3 rinses with the water to make sure that there's no any kind of contamination with it. I'm just gonna rinse that out 3 times.
And we've also got to rinse the cap from any contamination from prior water.
And then we're going to take a water sample, and then we took it into the lab. One of the experiments we were trying to see is if bacteria changes throughout the water column. So we're going to be plating it on a 96 well plate, and I'm going to put it into this little sample because we're going to be taking it with a pipette. We're about to deploy the wire flyer, the brainchild of Professor Chris Roman over there, and we've got it on the A-frame. A number of people involved because you're gonna lift this thing up. It's a little awkward, and then you've got a very heavy 2,000-pound weight that they've got to plop off the back of the ship. That's gonna pull a line taut, and then the wire flyer will get the A-frame will go back. The wire flyer is going to go in the water and go down there and collect some data.
Hi, I'm Ashley Maranzino. I'm a master's student studying marine biology at the University of Rhode Island. My research focuses on some of the sensory biology and some anatomy on some deep sea fishes. I'm not actually collecting anything on this cruise, just kind of getting an idea of sampling methodology that other scientists use to look at their research. So what we have for you today, we have some sargassum that we collected just overboard. So sargassum is a seaweed. So here you can see it's just a piece of seaweed. Seaweed is an algae, so it's basically a marine plant. So it's like a tree kind of for the marine environment. Obviously you can see not as big. but each of these little balls right here actually holds air so that the plant can float on the surface so that the sunlight can still reach it and the plant can still photosynthesize so it can still produce its own nutrients. So if you look, when you look at it within the ocean, you can see it floats on the surface, kind of like we have in this bucket. And what's really cool about these sargassum beds is that they float along the surface and a lot of little animals live within them. So they use the sargassum as kind of a little floating house to jump around the ocean when there's not a whole lot of other structures for them to live on. So over here we have some of the animals that are living in that water. So we have a couple of filefish. There are some shrimp, some crabs, some worms, so all sorts of little animals that live in the sargassum, and some of them will spend their entire lives living in a piece of seaweed floating in the middle of the ocean. And just to give you an idea of how these fish have adapted to live in their environment, here is a piece of sargassum. And here's another fish that typically lives in the sargassum beds, so a filefish. And then if we put it on sargassum, you can see how well the animal blends into its environment. So that really hides it from other fish that might be eating this guy in the environment, and allows it to kind of hide and camouflage into these sargassum beds as they float through the water.
Last thing we thought would be interesting to see the living quarters for the people who spend a lot of time on a ship. We're only here for 4 days. Sometimes they're up for 30 or more days at a time. So everybody's got different rooms down this hall, which you can see is quite spacious. And then if you have different shifts, you can just close yourself in so you can have privacy. So it's pretty tight, but surprisingly pretty comfortable. And it's not always clean, but that's the saltwater bathroom with the shower.
Sometimes I jump on the bike. My friend Joe's over there working out right now. He's gonna get a few more reps in, but I'm glad you came down to check it out.